Kelsey Zielinski, and I recently began as a PhD student with NC State's Plants for Human Health this past April. So today I'd like to talk to you about the work we've accomplished this summer for developing genetic and genomic resources to characterize the anthocyanin pathway in blueberry, and also how this ties into the long-term goals for my project, which is to improve the nutritional quality of blueberries even further. So first, though, I'd like to introduce my excellent interns. This is Claire Thetford of NC State University, Zoe Cantu-Backhouse of UNC Charlotte, Michaela Kidd of Pfeiffer University, and Joshua Marcus of NC State University. So I know that you learned a little bit about blueberries earlier, but blueberries are so highly regarded on this campus that we felt that they deserved two projects. So as Sam's group described earlier, blueberries are packed with secondary metabolites that are called anthocyanins, and these anthocyanins, when ingested, are known to actively scavenge the body and reduce free radicals by donating electrons to unpaired molecules. So this means that they act as antioxidants. And if these free radicals are left unchecked, then over time this can lead to a state of chronic inflammation in tissue, and this chronic inflammation often goes undetected until a disorder or disease surfaces. And while an acute inflammatory response is necessary for proper wound healing, chronic inflammation can often lead to conditions such as arthritis, asthma, heart disease, and certain types of cancers, and that's just to list a few. Fortunately, though, by consuming a healthy serving of blueberries along with a balanced diet, it's been shown that there is a decreased risk to acquire such ailments as I just mentioned. So even though we know this great things about blueberries and they have great impacts for our health, there are relatively few genetic resources that exist for this to study blueberry. So this leads me to the next part of my project, or the overall goal for this summer, like I said, which is to develop genetic and genomic resources to characterize the anthocyanin pathway in blueberries. So ultimately, we want to provide resources that could be handed off to a blueberry breeding program that could be used to increase the anthocyanin content in blueberries. And essentially, in a breeding program, there are two main components that you need. The first one being is to know how the trait you're interested in is inherited, so something like a genetic marker. And then you also need the germplasm, or plant that actually expresses the trait that you're interested in order to introgress that trait into the gene pool. So the two projects for this summer sort of fit in within those two categories. The first project is a way to develop a method that can be used to quantify anthocyanins and therefore produce a data set that could be used to obtain that genetic marker. So that is the first component we need. The second is essentially assessing a wild type of blueberry that is known to have pigment in their pulp. And this is exciting because if the color indicates that they could be expressing anthocyanins in their pulp. And currently the commercial cultivars do not express anthocyanins in their pulp. So one way to immediately increase the content of anthocyanins in blueberries is to have them expressed in the pulp as well. So the first project that is the methods-based project, and you might be wondering why we're developing a method when people have been quantifying anthocyanins in blueberries for years. Well, the way that the current method works is by weighing out a certain amount of blueberries, and you extract the anthocyanins from those blueberries, and that's regardless of the size of the berries. And as I just mentioned, anthocyanins are only expressed in the skin of commercial cultivars. So in the picture behind me, I've weighed out 6 grams of 3 different sized berries, and if you were to take the 6 grams of the smallest berries, you might find that that sample would actually have the highest amount of anthocyanins, which could lead you to believe that that perhaps is the biological basis, but in fact it's probably due to the actual method that you used. So, and there's actually some indication in the literature or documentation that there has been shown that there's a negative correlation between berry size and anthocyanin content. So we need to avoid that if we're conducting genetic studies where we want to map the genes that are related to anthocyanins. So the first question we have for this project is a bias actually introduced when using the whole berry method? And second, if there is this bias, then how can we remove it? So we've been working to develop and implement 2 new types of methods for this. So the second project is the look at the biochemical and bioactive profile of the Vaccinium staminium population that I was telling you about. And as I mentioned, these wild berries are sometimes they have a pigment in their pulp, and sometimes they do not. But if they're anything like the wild Alaskan berries that we're familiar with, then that could have great implications for human health, because the wild Alaskan berries contain such a range of anthocyanins that they express up to 26. So they are known to be an antioxidant powerhouse. But before we use these berries in a breeding program, we want to make sure first that they actually have, first we want to know, do they actually contain higher amounts of anthocyanins for purple pigment pulp than non-purple pigment pulp. We also want to know what are the differences in the actual types of anthocyanins. And then finally, what are the bioactive profiles between them. So now I'm going to turn it off to Josh, who's going to tell you about the first project, the methods and results, that is.
Thank you, Kelsey. As Kelsey was saying, one of the primary goals this summer was to establish whether or not the traditional whole berry method of anthocyanin sampling introduces a size bias. Basically, how we went about this was we harvested a number of genotypes. We then weighed them out into samples, took photographs of the respective samples, and then performed extractions. The photographing element was where the majority of my efforts went into this summer. We were provided with a piece of code called GINA from some colleagues at the University of Wisconsin at Madison, which was originally designed to extract the dimensions of cranberries, but I modified it to extract the dimensions of blueberries. And then with these dimensions, I transformed them into surface area and volumes, and made surface area to volume ratios, remembering that small berries have very high surface area to volume ratios, while large berries have low surface area to volume ratios. And then I correlated the anthocyanin contents to the surface area to volume ratio and made a plot, as you can see in our results. As we expected, we see a negative correlation between size and anthocyanin content. And to talk about some of the other methods of anthocyanin sampling we developed this summer, I'm going to pass it off to my colleague, Michaela. Here you go, Michaela.
Thanks, Josh. So as Josh was saying, we see a negative correlation between anthocyanin content and berry size within the whole berry method. So this summer, we began working on new methods we could use to accurately quantify anthocyanin content while removing this bias. So the squeeze method, essentially what is done is you cut the berry in half, you squeeze it to separate the pulp and the skin is used for extractions and HPLC analysis. We implemented this method on a subset of a mapping population whose parents were chosen for differences in flowering times. So since they were chosen for differences in flowering times, we needed to make sure that the progeny or the population we would use were segregated for anthocyanins. So for our future work, we will be able to map genes related to anthocyanin production and anthocyanin production in the flavonoid pathway. So after we get our results, we see that there is a 4-fold difference between the individual with the lowest overall anthocyanin content and the individual with the highest overall anthocyanin content. And that was to be expected because there's also a 4-fold difference within the parent's anthocyanin content. So it leads us, we can conclude that the population is segregating for anthocyanins, and we will proceed in the future with this work. So to answer questions about Project 2, I will turn it over to Zoe and Claire.
Thank you, Michaela. So my part in this project was determining the biochemical profiles within the staminium berries. As Kelsey just mentioned, there are 3 main questions that we need to answer with this project. The first one being the total amount of anthocyanins within the staminium berries. As you can see on the graph, there is a higher amount of anthocyanins within the higher pulp within the staminium berries, as compared to those staminium that has less pigment within their pulp. This then leads us to our second question, which is the different amount of anthocyanins within the cultivated berries along with the staminium. As you can see within this chart, there are two main colors, blue and green. The blue represents the cultivated berries as the green represents the staminium berries. The blue has a significant amount of anthocyanins. As a matter of fact, it has 22 different types of anthocyanins within them as compared to the staminium, which only has 5 different types of anthocyanins within those berries. With that being said, I'm going to pass along to my colleague Claire, who will answer question 3.
Thanks, Zoe. So my focus this summer was mainly on the cell culture aspect of this project, which directly answers question 3, which compares the Vicinium-Staminium lines, which have the purple pulp, and their bioactivity versus those who do not have purple pulp. And for our methods, we first separated the skin and the pulp to create a raw extract to be used in a nitric oxide assay. And this assay, you first have to cause inflammation within macrophage cells using a chemical called LPS. And once these cells are fully inflamed, we then treat it with a positive control DEX, as well as the several extracts that we created separating the skin and pulp. And to put it into comparison, we do want a decrease in nitric oxide concentration because that directly correlates with the efficiency of the extract, decreasing the inflammatory response within our cells. And so after running these assays, we then put our results on the following graph. And as you can see here, there are minor differences between the staminium, skin, and pulp varieties, but as you can also see, there is not that much of a significant decrease with our positive control DEX. And with these minor statistical differences, we kind of concluded that it wasn't due to the biological activity of the extract that caused these minor differences, but rather our methods. And so we're hoping that with the change in our methods, we can find greater results and also what we expect to see in the positive control of the DEX. And now I'm going to turn it over to Kelsey, who's going to conclude our presentation.
Thank you, Claire. So just some updates for the future work for the first project, which is the method development. In the future, we want to increase the number of samples that we used in testing whether the whole berry method had any bias. We've actually harvested over 50 different genotypes to test this, but with time constraints, we've only had a chance to run 15. Also, with the mapping population Michaela discussed, since we are seeing that they are segregating for anthocyanins, then we'll follow up and use this population to map QTLs that could be related to anthocyanin production. For the second project of the purple pulp berries, we'd like to canvass research stations to find more lines of staminium that we could use to include in our sample population and see if there are any other differences that exist. And then we also need to refine the protocol used for the nitric oxide scavenging assay. As Claire mentioned, it was probably due to the lack of statistical differences, probably due to something in our protocol. So for example, in the future, we'll probably need to increase the concentrations of extracts we use to actually get a proper response. And if you have any other questions or just want to come talk to us, please come see our posters. They're number 4 and number 10. And in summation, thank you so much for your attention.